everyone, this video is all about citric acid cycle. And the learning focus is for you to be familiarized with the 8 reactions that constitute the citric acid cycle and describe the overall chemical transformation that occurs in one round of the citric acid cycle. And we can define citric acid cycle as the series of biochemical reactions in which the acetal portion of acetyl-CoA is oxidized to carbon dioxide gas and the reduced coenzymes flavine adenine dinucleotide and nicotinamide adenine dinucleotide are produced. So the citric acid cycle gets its name from the first intermediate product in the citric acid cycle, which is the citrate. But remember, even though the first intermediate product is the citrate, they still call this process a citric acid cycle because the citric acid at cellular pH level is also known as citrate. So, but they prefer to call it a citric acid cycle, and this one is also known as Krebs cycle after its discoverer, Hans Adolf Krebs. And this is also known as tricarboxylic acid cycle in reference to the 3 carboxylic groups present in citric acid. The citric acid cycle occurs in mitochondrial matrix, and we will have the process of oxidation in which it will produce the reduced form of nicotinamide adenine dinucleotide and flavine adenine dinucleotide. And you will encounter that in the 4 of the 8 steps. And we also have here the carboxylation process, wherein a carbon chain is shortened by the removal of a carbon atom as a carbon dioxide gas molecule. You will also encounter that in the 2 of the 8 steps here. And this is the overall reaction of the citric acid cycle. The first thing that will happen is the acetyl-CoA from the second stage of the biochemical energy production will produce a citrate. So that will occur in step 1. So this is the reaction for that. For step 1, the formation of citrate. So the acetyl-CoA from the second stage of the biochemical energy production will react with the oxaloacetate. You can obtain oxaloacetate from the digestive enzymes that can be found in the cell. So with the help of the enzyme citrate synthase, in which it will combine the two and will rearrange the sequence of the bond. So I highlighted here the green parts for you to be focus on that. So here the acetyl-CoA will be placed at the top, and as you can see here we have the subscript 3, but here we only have 2 because the 1 hydrogen will serve or will be placed here. You have no hydrogen here at the oxaloacetate, but here in the combination of 2 the hydrogen, another hydrogen will be placed here. That is why you have only a 2 as subscript here instead of 3. And then this will be known as citral-CoA, and the process they undergo is what they call the condensation process. And another step will occur in which we will have the hydrolysis of the Thioester bond. This is the Thioester bond, right? As you can remember from the previous lecture, Thioester bond is the bond that connects the sulfur and the carbon in a acetyl-CoA. So it will hydrolyze that, meaning to say it will break the bond. So the S-CoA here, the sulfur coenzyme A, will be removed in the process, and the enzyme responsible for that again is the citrate synthase. With the addition of water, we will be using water here. So if you will use the water, you will be able to break the bond here. Unlike if you will only use the citrate synthase, you will just arrange the structure of the molecule. But here, since you used water, you will be able to break the bond of that. And after you break the bond, as you can see, we have here the C double bond O. But once you remove that, since there is an excess bond for the carbon, that will be expounded as COO or carboxylate ions such as this one. And what will happen next, the water that we used here the 1 molecule of the hydrogen will be used here in the S-CoA here that will serve now as coenzyme sulfhydryl. Another hydrogen was placed here and a free hydrogen ion. Now, the oxygen here, the remaining oxygen will be attached here, the COO. Okay, that is why you no longer have the double bond here because you now have the carboxylate ion at the top. So you have now here as the product, we have the citrate and the coenzyme sulfhydryl plus the hydrogen ion. But the main product for the step 1 is the formation of citrate. And then next we have here the step 2, formation of isocitrate. So the enzyme responsible for this, we have here the citrate from the first step. So the enzyme responsible is the aconitase, and aconitase will remove the water here, water molecule, and you will have an water molecule that is being expelled in the citrate. That is why you have a outward arrow here denoting the expulsion of the water molecule. And if you remove that, you will have now the cis aconitate. When you encounter a word cis, it meaning to say the molecules are all aligned in single place, in one place. Unlike for trans, they are in opposing sides. So the water that has been expelled from this molecule will be used again with the help of the enzyme aconitase. Since this is an isocitrate, from the term itself, we will be forming an isomer of the citrate. So the purpose of this enzyme is to isomerize the citrate. So as you can see here, the hydroxyl is placed at the third carbon and the hydrogen is placed at the fourth carbon. But here, on the use of the water that will be used again in the process using with the help of the enzyme aconitase, it will reassemble that, in which the hydrogen is now placed at the third carbon and the hydroxyl group as placed at the fourth carbon. So that is what we mean by isomerization. Same chemical formula, but they have the different arrangement. So why same? Because you have here 1, 2, 3, 4, 5 carbons here as well. And you have how many hydrogen? 1, 2, 3, 4, 5. You have here 1, 2, 3, 4, 5. How many oxygen? 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6, 7 as well. So they have the same chemical formula, but they differ in the arrangement. That is why in step 2, it's all about the isomerization process. And now we have here the isocitrate, the isomer of the citrate. For step 3, the process is all about the oxidation of isocitrate and the formation of carbon dioxide gas. So this is the isocitrate from the step 2 here. And with the help of the enzyme isocitrate dehydrogenase, from the term itself, dehydrogenase, it will remove the hydrogen in the isocitrate. And with the help of the coenzyme nicotinamide adenine dinucleotide, the oxidized form, the hydrogen here, it will reduce that, and it will produce the reduced form of nicotinamide adenine dinucleotide. And then free hydrogen ion, that is where the hydrogen here went. Okay, dyan napunta dyan. Dito yung hydrogen, and the other hydrogen is here. And then, after you remove the hydrogen here, you will have now the oxalosuccinate. Okay. And what will happen next is the carboxylation. If this is oxidation or the removal of the hydrogen here, we have here the oxalosuccinate, and the next step that will occur is what we call the decarboxylation process, or the removal of the carboxylate ion. So we will be removing this one, and the hydrogen ion from here will be used in this process to attach the hydrogen ion here that will undergo the process, will attach itself on the hydrogen here instead of a COO here that will be removed, and you will have here a carbon dioxide gas plus CO2. Okay. So carbon dioxide gas will be produced here. You have now here the hydrogen from the hydrogen ion, and COO will be exhaled in a breathing process. Carbon dioxide gas that we are exhaling during the breathing process came from this step, or from the citric acid cycle. And then what will happen next? You have now the alpha ketoglutarate. This is now the enzyme that you have plus a carbon dioxide gas. And then what will happen next is we have here I have mentioned earlier the cis and trans isomers from the step, I think that's step 2. Yes, step 2. So when we say cis, the elements of the molecules are in the same place or at the same side, while in trans isomer, that is, they are in the opposite sides. And then this is very common in nature, and this one is rare in nature, and here are the other definitions. Now, for step 4, which is the oxidation of the alpha ketoglutarate and the formation of carbon dioxide gas, you have here your alpha ketoglutarate. And don't be confused because I just compressed the CH2 here. As you can see, the CH2 here is expounded, HH, and then I just write it in a way as CH2. So you have now here your CH2 here, but this is also an alpha ketoglutarate. So we will be using the NAD plus the hydrogen ion plus you will obtain that from the step 1. We will be using the byproducts here, coenzyme sulfhydryl and the hydrogen ion. So that is here for step 4, this one. Okay. We will be using the oxidized form of the nicotinamide adenine dinucleotide plus the hydrogen ion and the coenzyme sulfhydryl from the step 1. And with the help of the enzyme alpha ketoglutarate dehydrogenase complex, we will be removing the hydrogen here in the H, and the NAD will be reduced into NADH+, such as this one. So you will have here now the NADH, the reduced form of nicotinamide adenine dinucleotide, plus the another hydrogen ion from here, since you also use this hydrogen ion here. And then there will be also the carboxylation process, or the removal of the carboxylate ion. So when you remove this one, you will be attaching the sulfur coenzyme A here at the bottom. So you will now have here the succinyl-CoA or succinyl coenzyme plus the reduced form of nicotinamide adenine dinucleotide plus the carbon dioxide gas from the COO here, or from the carboxylate, and then plus a hydrogen ion, free hydrogen ion. Okay, since you only used one hydrogen from the removal here, in which you produced a reduced form. And that is for step 4. You have here the succinyl-CoA as product. And then for step 5, you will be having the thioester bond cleavage in succinyl-CoA and phosphorylation of guanine diphosphate. So from here, the thioester bond cleavage, it means to say that we will cleave the bond here. Thioester bond is the bond that connects the sulfur into another carbon. And when you say cleave, it will cut off or it will split the bond. So, puputulin yung bond na yan. And then, it will be using the phosphorylation. We will be using this HPO4 acid here to use the inorganic phosphate. So, when you cleave the bond of the S-CoA, you will be having a phosphate. An oxygen here. And then, the phosphate or inorganic phosphate here. So you now have the succinyl phosphate, and the sulfur coenzyme A here will attach itself on the hydrogen here. So then it will produce a coenzyme sulfhydryl again. It will that will be released. And the enzyme responsible for this is the succinyl-CoA synthetase. So the difference between succinyl-CoA synthetase from the synthetase does not use the energy, but this one it uses an energy. It will be using the guanine diphosphate. So this guanine diphosphate will be released as JTP or guanine triphosphate because the phosphate here, this one, will be removed. This one. So what diphosphate, meaning to say 2 phosphate plus 1 phosphate here, you will have 3 phosphate. That is why you have guanine triphosphate here. And the enzyme that catalyzes this reaction is succinyl-CoA synthetase. So if you remove this one, you will be able to expound this already. So this CO here will be a COO because you are able to break the bond here. So that will be a carboxylate ion, COO, just like this one. And you will have now here the succinate. Phosphorylation of JDP, when you phosphorylate that, you will have a JTP here. That is the one of the important phenomenon in step 5. Next step is step 6, or the oxidation of the succinate. When we say oxidation, that is, again, we will be removing a hydrogen ion here. So the coenzyme that will catalyze this one is the flavine adenine dinucleotide, the oxidized form. And we also have here the succinate dehydrogenase that will remove the hydrogen here. So it will remove 2 hydrogens from one here and another one here. So it will be therefore reassembled into another structural form. You will have here a double bond. You will have a double bond because of the breakage of the 2 hydrogens. And 1 hydrogen will be placed, they will be configurized into another form. The hydrogen will be placed here, and another hydrogen will be placed here, and this is the trans form of the fumarate. Trans. Okay, because we have the cis form of the fumarate. The cis form of the fumarate is toxic and lethal to the digestive system. It will cause you death and complications in your digestive tract. The cis form. But the succinate dehydrogenase only produces the trans fumarate, this one. This is the only configuration that they produce. So we remove 2 hydrogens, and the 2 hydrogens will be the reduced form of the FAD. It will be added here. So you will have now the reduced form of the flavine adenine dinucleotide. That is for step 6. We have here the end product, the trans fumarate. What is the look of the cis fumarate? We have here the carbon double bond, carbon, and then the COO is placed at the same side. and the hydrogen is placed at the same side as well. This is the cis form of the fumarate, and we call this as maleate. Maleate. This is toxic to our body and can cause to fatal death. And then we have here step 7, hydration of fumarate. We will add water on the fumarate, and if you will add water on that, so this is the product we have here, and this is the same. They are in opposing side, the hydrogen, and with the help of a water and the enzyme fumarase for the hydration, the HO or the hydroxyl of the water will be placed at the second carbon, and the another hydrogen will be placed at the third carbon, and you will now have the left-handed malate. Now, for step 8, we have here the oxidation of left-handed malate to regenerate oxaloacetate. This is the left-handed malate from here. And then the NAD+, the oxidized form, and with the help of the malate dehydrogenase, we will be removing the hydrogen here. And therefore, if you remove that, you will have a double bond here for the carbon, because you have two placed here. And then the hydrogen, the removed one hydrogen will serve as the reduced, will be added here in the oxidized form of the NAD+, and you will have the reduced form, nicotinamide, adenine dinucleotide, plus a free hydrogen ion. That is where the two hydrogens went. And you have your product now. We have here the oxaloacetate. This is all about regeneration of oxaloacetate. It will regenerate the first reactant. Rather, this is the reactant of the first step. So, to summarize the process in the citric acid cycle, you have here the acetyl-CoA from the second stage, and with the condensation process, it will produce the citrate, condensation and hydrolysis of the thioester bond, and then with the isomerization process, it will isomerize the citrate into isocitrate, and with the oxidation and the carboxylation, it will produce an alpha-ketoglutarate. Then, with the oxidation and the carboxylation again, it will produce the succinyl-CoA. And phosphorylation, it will produce a succinate. Then oxidation, it will produce a fumarate. Then hydration, it will produce a malate. Then oxidation, it will produce the oxaloacetate. This is the process of that. So, as you will look back here in the cyclic diagram of the citric acid cycle, you have here the products, the isocitrate, the alpha-ketoglutarate, the succinyl-CoA, the succinate. You have here the fumarate, the malate, and the oxaloacetate. So, this is a cyclic process. And then, what else? So, the essence of the citric acid cycle is for you to know the end products. You can use or you can create a mnemonics. For me, I have here, can I ask some super fantastic memes on Krebs cycle? So, the C stands for citrate, the first product of the step 1. Then, you will have isocitrate, the second product from step 2. Then you will have an alpha-ketoglutarate from step 3. Then you will have a succinyl-CoA from step 4, succinate from step 5, fumarate from step 6, and malate from step 7. You will have the oxaloacetate, another product that will be combined to acetyl-CoA on step 1. This is the step 8 here. So, you can make mnemonics for you to understand better the end products or for you to be familiarized with the end products of the Krebs cycle. So, that is all for Krebs cycle.